Hi, my name is Brooke Preston, and in this video I will explain what gene expression is and the processes involved in expressing a gene. After that, I will demonstrate translation in a stop-motion video that I have created. So first off, what is gene expression? What are the processes involved, and why is it important? Well, gene expression is the process by which information contained in a gene is transcribed and translated into an essential product, such as a protein. So you remember that genes are made up of DNA. In the first part of gene expression, the information found in a strand of DNA is used as a template to create mRNA. This process is called transcription. Once that strand of messenger RNA is complete, it can be used in translation. It is easy to remember what step this is called because in the process, the nitrogenous bases in mRNA are being translated into something new, the amino acids. An important thing to note about translation is that mRNA, tRNA, and rRNA are all being used in this process, but rRNA is part of the ribosome. So I have modeled mRNA and tRNA, but did not specifically model and label rRNA because it is part of the ribosome that I have included. So now, to give you a better understanding of translation, I will show you the video of what the process actually looks like. In the nucleus, DNA is used as a template to make messenger RNA, or mRNA. Then, in translation, the specific information contained in mRNA is used to make a polypeptide. Nitrogenous bases are grouped together into three-letter codes called codons. Codons code for specific amino acids. There are also 4 special codons. One that is found at the beginning of the mRNA strand that codes for start, AUG, and 3 others found at the end that code for stop. Translation begins when the small ribosomal subunit binds to the mRNA strand at the start codon. Each amino acid is brought to the ribosome by a specific tRNA, or transfer RNA. The type of amino acid is determined by the anticodon sequence of the tRNA. Complementary base pairing occurs between the codon of the mRNA and the anticodon of the tRNA. Uracil pairs with adenine and guanine pairs with cytosine. After the initiator tRNA binds to the start codon, the large ribosomal subunit binds to form the translation complex. During elongation, amino acids are brought to the mRNA strand by a tRNA molecule through complementary base pairing of the codons and anticodons. Each anticodon of a tRNA molecule corresponds to a particular amino acid. A charged tRNA molecule bonds to the A site, and a peptide bond forms between its amino acid and the one attached to the tRNA at the P site. The complex slides down one codon to the right so that the uncharged tRNA can exit from the E site, and the new A site is now ready to accept another tRNA molecule. This process continues until a stop codon is reached. Once the polypeptide chain is formed, a release factor will bind to the stop codon at the A site, and the polypeptide will be released from the tRNA. And that was my explanation of protein synthesis.